So, brain metabolism and fasting. So, the authors of this particular study have noted how the importance of glucose when it comes to energy yield as an energy yielding substrate. Glucose is involved in many of the processes to maintain optimal brain function. Glucose is crucial for the brain's many metabolic pathways. Glucose is usually readily produced, so it's in large amounts in order to maintain these functions. However, during fasting, glucose must be drawn from limited carbohydrate stores, from amino acids and from proteins, and other precursors such as glycerol and lactate. However, these limited stores do not produce enough glucose in order to maintain optimal brain function. So, how is it that humans are capable of surviving prolonged starvation when these protein and carbohydrate stores that are required for gluconeogenesis to maintain functions are limited? That is what the authors of this study are looking into. So, the authors examined these three obese subjects, and they were administered with a particular diet with an intake of multivitamin capsules, 17 milliequivalents of sodium chloride, and 1,500 milliliters of water each day. Now, this was a period of time where they were engaged in a fast, which lasted 38 to 41 days. After this, the subjects were subjected to catheterization and many other uh, tests to analyze things such as the metabolic substrates in their bodies that could probably could answer why it was sufficient to have this low amount of glucose and still be able to function. Obviously glucose concentration was also measured and whatnot. And so as you can see here, the calorimetry, blood flow, blood, and urine analysis were performed. Obviously, the subjects were also pre-medicated and administered with 100 milligrams of sodium pentobarbital, 50 milligrams of pyridine sulfate intramuscularly before any of these analyses occurred. Now, the results of this particular study showed diminished daily glucose production in subjects, as was expected. Now, however, there was an influx or an increase in beta hydroxybutyrate and acetoacetate, which replaced glucose. So, in the absence of glucose, these keto acids came in and took the reins to providing the energy yield that is sufficient to maintain optimal brain function. So glucose is no longer the brain's primary means of attaining fuel at the point of starvation. So there's also a low respiratory quotient, meaning that CO2 was not, there was a decrease in CO2, suggesting a carboxylation mechanism that exists that is not yet accounted for by this experiment and the manometric techniques used in this experiment. So the techniques used were not able to detect CO2. In conclusion, though, keto acid utilization, in addition to gluconeogenesis involving the fat products, which includes the carbohydrate stores and whatnot, was sufficient enough to satisfy the central nervous system, unlike what was previously thought. So the missing link was the keto acids, allowed for a minimal amount of glucose to be produced, but and that was enough for optimal brain function. So the keto acids were the missing key. Predictions, well, to be honest, I don't believe that the data that we got from this particular study is enough to be generalizable to the general population. This was in a very the sample was a very limited sample. It was also a sample that was pretty extreme of obese people. Now this might not be generalizable to the average Joe or and whatnot. So there might be different metabolic pathways, different effects on the metabolic pathways in someone who is not obese. And yeah, that is all.